Good morning, grade 9 students. In this session, we're going to go to the digestion revision. So we're going to talk about digestive system. We're going to talk about the enzymes. We're going to talk about the different food tests that are found in the lesson about or the chapter about digestion. You have to know what's happening, and we're going to review some of these notes together. These notes already you have covered them before, so this session is a revision session. Here we go. First of all, let's talk about Fehling test. What is the aim of the Fehling test? Fehling test is done to identify or to test the presence of sugar, reducing sugar. All the sugars in our body, they are reducing except for sucrose. So all of them, they we used with them this test is called the Fehling test. Unless sucrose. Sucrose is a non-reducing sugar. So all the tests or the sugars are reducing sugars except for sucrose. So why do we do the Fehling test? Fehling test is done to identify a presence of reducing sugar, meaning that all of the sugars unless sucrose. What is the procedure? What are the steps? Put a piece of food in a test tube, add equal drops of phenol A and phenol B in a solution, and the solution will be heated over the Bunsen burner over the source of heat. The phenol solution has blue color. If we have a positive result, meaning that we have reducing sugar, so the color will be brick red precipitate as shown here. We have a brick red. However, if it's a negative result, there is no change in color, so the color will remain blue. So this is the test, the first test, which is the Fehling test. So Fehling test is done to identify the presence of reducing sugar. First of all, we have to heat the food in the test tube, add few drops of phenol A and phenol B. If there is a brick red precipitate, this is a positive result. If there is no brick red precipitate and the solution remains blue, this is a negative result. What does the positive result indicate? It indicates the presence of reducing sugar, while the negative result, it indicates the absence of reducing sugar, where the solution remains blue in color. So the first test is the Fehling test. I repeat, Fehling test is done to identify the presence of reducing sugar. All the sugars they are reducing except sucrose, which is a non-reducing sugar. And if it's a positive result, we will have brick red precipitate. While if it's a negative, the solution remains blue, indicating the absence of reducing sugar. Moving now to iodine test. Iodine test is done to identify the presence of starch. Here the iodine test, as you see, it has a brown, orange, yellow color. Whenever it's added on the starch, it will give this color, which is dark blue color. So dark blue color if we have a starch. If we don't have starch, the solution will remain brown, orange, yellow in color. So here, this is the second test. Iodine test is done to identify the presence of reducing sugar. What is the procedure? Put a piece of food in a small dish, add few drops of iodine solution. It has brown orange color. Sometimes they say it's yellow orange color. It's okay. So brown orange color. Here the results. If it's positive, we will have a dark blue color appears. Dark blue color indicates the presence of starch. However, the negative result, it's no change in color. The solution remains brown orange. So this indicates the absence of starch. So I repeat the iodine test. Iodine test is done to identify the presence of
Starch.
starch. Very good. So here, if it's a dark blue color, meaning that we have starch. If there is no dark blue color, meaning that there is no starch, if the solution remains brown orange in color. So my first question is, why do we do a first fehling test? What is the aim of fehling test?
Yeah. Okay, the failing test.
Yes.
Okay, it's done. What the aim of the failing test is done to identify the presence of
Reducing sugar,
hmm,
right?
of starch.
Starch. Mm. Abbas, reducing
Yes.
sugar. Fehling test is done to study the presence of reducing sugar, right?
Yes.
Mm. So, the Fanning test is done to identify the presence of reducing sugar. However, the iodine test is done to identify the presence of starch. These notes, they are very important. If we didn't study them, we can't solve any digestion exercise. This is very clear. So I'm going to be very clear from the beginning, as I told you from the beginning of the year. If you didn't study these notes, you are not able to solve any digestion exercise. This is very clear. Moving now to the third test. It's the Bayer test. Bayer test is done to identify the presence of proteins and slash or polypeptides. So proteins, polypeptides, or even both of them. The procedure is as follows. Put a piece of food inside the test tube. This is the test tube, we put a piece of food. Then we add equal volumes of copper sulfate and sodium hydroxide. This is the copper sulfate, CuSO4. Sodium hydroxide, NaOH. For example, we add 2 drops of NaOH and 2 drops of CuSO4. Then the formed mixture will have a blue color. It's called the Bayer solution. It has blue color. So here we want to test the presence of proteins and or polypeptides. So the positive results, a violet color appears, indicating the presence of proteins and or polypeptides. While if the solution remains blue in color, this indicates there is no change in the color. It remains blue. So this means that the absence of proteins and or polypeptides. So here are the positive results. It's the purple or the violet color, meaning that we have proteins and or polypeptides. However, the negative results is that the
Sure?
solution remains blue in color. So this solution remains blue in color. So this is the negative results. So here I will repeat. So the Bayer test is done to identify the presence of proteins and or polypeptides in food. This is the aim of the Bayer test. So the solution has blue color at the beginning. So it has blue color due to the copper sulfate. And then we move to the results. If we have a positive result, meaning that we have proteins and or polypeptides, so this means that the solution or this food contains proteins and or polypeptides. However, if we have no change in color, and the solution remains blue in color, this means that we have no proteins and or polypeptides. Moving to coagulation test. Coagulation is done only for proteins. So if the food coagulates, if the food shrinks like this, if the food coagulates, it's liquid, then it coagulates in the case of eggs. So here we have proteins. What do we do? Simply heat the food till boiling. If the food coagulates, so this is a positive result and it contains proteins. However, if there is no coagulation, no proteins. So what's the difference between the Biret test and the Fehmi? Biret is for both proteins and or polypeptides. Why coagulation? It's only for proteins. Here we have four tests till now. Let's review these tests before we move to the new test. So first we talk about the Fehling test. We said it's done to identify the presence of reducing sugar, which are all the sugars except sucrose. And moving now to the positive result of the Fehling test, it gives a brick red precipitate. However, the negative result, the solution remains blue. While the iodine test, which is number two, is done to identify the presence of starch. If we have dark blue color, meaning that we have starch. If there is no dark blue, meaning that there is no starch. So here we have two tests. Failing test for reducing sugar. It gives brick red precipitate if it's positive, and it remains blue if it's negative. The Biret test, it gives dark blue color with the starch. If there is no starch, the solution remains brown orange in color. Test number three, it's the Biret test, which is done to identify the presence of proteins and or polypeptides. If the food gives a violet color or purple, this means that we have proteins or polypeptides and or polypeptides. However, if there is no proteins and or polypeptides, the solution remains blue. Number four, it's the coagulation test, which is done to identify the presence of proteins. Procedure, heat the food till boiling. If the food coagulates, this is a positive. If there is no coagulation, this is a negative. Moving now for the translucent test. Translucent spot test. It's done to identify the presence of lipids. One example about this is the oil. If you put a drop of oil on this paper, if it gives this translucent spot, this means that we have a positive result and we have lipids. However, if there is no translucent spot like the case of water, so here the solution contains no lipids. So this is a negative result. So here, let's repeat it together. Translucent spot test is one of the tests done to identify the presence of lipids. If there is lipid, it will give a translucent spot after rubbing the food on a piece of paper. While if there is starch, if there is lipid, sorry, it gives a translucent spot. If there is no lipids, there is a negative result, and there is no translucent spot. Moving for sodium test, which is done to identify the presence of lipids. We have to add sodium test solution. If there is a pink color, so this means that we have lipids. If there is no pink color, meaning that there is no lipids. So as you know, digestive system is divided into both digestive tube and digestive glands. You have to know that digestive tube, they are the organs through which the food passes through, wherever the digestive gland organs through which the food doesn't pass through. You have to know what are the organs of the digestive tube. They are the mouth, the esophagus, the stomach, the small intestine, the large intestine, and finally the anus, where the food is excreted and released in the form of waste or stool. However, the digestive glands, we have the salivary glands, the gastric glands, the pancreas, the liver, and the intestinal glands. You have to know what is the function of the digestive glands. Digestive glands, they have an important function. They secrete digestive juices to their site of action. So this is the function of digestive glands. You have to know that digestive glands, they secrete juices and their sites of action. For example, salivary glands, what do they secrete? They secrete saliva. Where? Inside the mouth, where the medium is neutral. Let's see if you remember. So what is the neutral medium? The pH is equal, greater than, or less than. Let's see by raising your hands, please. Neutral medium, the pH is equal to what, or greater than what, or less than what? Yes, Fadil Basma, neutral medium. What do you think about the pH of the neutral medium? Yes,
Equal
Fadil?
7.
The
7.
7. What if it's acidic?
More than 7.
More than 7. Very good. Very good. Yes, Kalam. What do you think when it's acidic? Thank you, Fadil.
The acidic would be below 7, like under 7
Below
pH.
7. Very good. So if it's acidic, it's less than 7, ya Fadil. While if it's basic, it will be greater than 7. So here, you have to know this. So here in the gastric glands, they secrete gastric juice where in the stomach is the site of action, where the pH it's less than 7. In the small intestine, the medium is basic, where the pH it's greater than 7. So the pancreas, it secretes what? The pancreas, it secretes pancreatic juice in the small intestine, where the medium is basic. The liver, it secretes bile, where also the small intestine. The intestinal glands, what do they secrete? They secrete intestinal juice, where the small intestine, where the medium is basic. So from now on, you have to know the following. The salivary glands secrete saliva in the mouth. Gastric glands, it secretes gastric juice in the stomach. And the pancreas secretes pancreatic juice. The liver, it secretes bile. And the intestinal glands, they secrete intestinal juice, all of them in the small intestine. So here you have to know these notes. And medium, acidic, basic, or neutral. We said that neutral, it's around 7, between 6.5 to 7.5. This is the neutral. Greater than 7, it's basic, meaning that this one, the pH 14, is the most basic one, while less than 7, the one is the strongest acid here. So as we move away from the neutral to be the strongest acid or the strongest base. Moving now to talk about the digestion. What is digestion? Digestion is the set of both mechanical and chemical transformation of food into simple nutrients to be used by the body cells to produce the energy needed for the cellular activities. So here we have to know that the digestion it includes both chemical and mechanical digestion, where it digests food, complex food, into simple nutrients. You have to keep in mind that all the juices they contain enzymes except for bile. Bile doesn't contain any enzyme. What are the enzymes? The enzymes, they are the biological scissors. They cut, they digest, they hydrolyze, they break down, they transform. All of them, they have the same meaning. They digest food. They speed up the reaction. They make it faster, but remain unchanged at the end of the reaction. So all the digestive juices, like saliva, like the gastric juice, like the pancreatic juice, the intestinal juice, all of them, they contain enzymes, unless the bile, which is secreted by the liver. So the bile, which is secreted by the liver, contains no enzyme. So there is no enzyme in the bile. Moving now to this table. This table, I asked you before to study, but first, this week, I don't want you to study this, to review this. You have to review the another table that I have already uploaded. So first, you have to know that the organs, we have the digestive gland, digestive juice, enzyme, the substrate, the product, the medium, and temperature. So first, inside the mouth, what do we have? We have which digestive gland? We have the salivary gland. So the salivary glands, it secrete a juice. It's called saliva. Inside the saliva, we have a special enzyme. This enzyme is called salivary amylase. So salivary amylase, it works on what? Which substrate? It works on starch. To give what? To give maltose. So substrate is the substance that the enzyme is working on. So here the salivary amylase, it works on starch to give maltose, which is reducing sugar, and the medium is neutral. So the pH is equal to 7, approximately equal to 7, and the temperature is 37 degrees Celsius. Here we talk about the first digestive enzyme, first enzyme, which is found at the level of the mouth. So here is the first enzyme. So the first enzyme is the salivary amylase that works on starch to give maltose. Medium is neutral, and temperature is 37 degrees Celsius. Now we move to the stomach. This is the saliva inside the mouth. We talk about it. This is the saliva. Saliva that's found inside the mouth. This is the saliva. Okay? So the saliva contains the enzyme salivary amylase that works on starch to give maltose. Second enzyme in the stomach. So the stomach, we have the gastric gland. The gastric gland, it secretes the gastric juice. So the gastric juice contains the enzyme pepsin. Pepsin as the Pepsi. Pepsin is acidic. So pepsin, it works on protein to give peptide. So the triple P, pepsin, protein, peptide. So always, whenever you hear the pepsin, it's acidic. Why? Because the Pepsi, like Pepsi. So pepsin is like Pepsi. Pepsin, acidic, and the pH it's less than 7. And the temperature always is 37 since it's the temperature of the normal functioning of the enzymes. So I repeat this. In the stomach, we have the gastric gland. The gastric gland, it secretes a juice which is called a gastric juice. This gastric juice is containing the enzyme pepsin. So pepsin, it works on protein to give peptide, PPP. Pepsin works on protein to give peptide. The medium is acidic and the temperature is 37 degrees Celsius. So here, first two enzymes, salivary amylase in the mouth works on starch to give maltose, medium neutral, temperature 37. The second enzyme was the pepsin that is found in the stomach inside the gastric juice. The pepsin works on protein to give peptide. Medium is acidic and temperature is 37 degrees Celsius. And moving now to the small intestine. Did you see here? Only the mouth is neutral, and only in the stomach it's acidic, while the rest, all of them, they are basic till now. So only these they are different. So salivary amylase it works on in a neutral medium, while pepsin it works in acidic medium, and the rest all of them they are basic. Okay, so here you have to know that the third in the small intestine, the third organ, the pancreas, it secretes a special juice which is called pancreatic juice that contains the enzyme peptidase. Peptidase, it works on peptides to give amino acid, where the medium is basic and the temperature is 37 degrees Celsius. So here, pancreas secretes a juice called pancreatic juice, contains the enzyme peptidase. It works on peptides to give amino acid. Medium is basic, temperature is 37 degrees Celsius. Now, moving to the pancreas also, secreted juice called pancreatic juice contains an enzyme called trypsin. So here trypsin, it works also on protein, but that doesn't give protein, it gives amino acid, which is the simplest form of protein. And the medium is basic, temperature is 37 degrees Celsius. And moving now to pancreas, secretes pancreatic juice containing the enzyme amylase. And here we have amylase, and the mouse have salivary amylase. Both of them work on starch. Both of them they produce maltose. But the amylase here, the medium is basic, while the salivary amylase, the medium is neutral. So both of them they work the same function, but different mediums. One of them, salivary amylase, is in a neutral medium, and the amylase is in a basic medium. While moving for pancreas, it secretes a juice called pancreatic juice that contains an enzyme called lipase. Lipase, as its name indicates, lipase, it works on lipids. Lipase works on lipids to give fatty acids and the glycerol. The medium is basic, where the pH is greater than 7 and the temperature is 37 degrees Celsius. Moving now to liver. So liver, it secretes a juice, it's called bile. As we said before, bile contains no enzyme. Bile, it works on lipids to give emulsified lipids in a basic medium, temperature 37. What are the meaning of the emulsified lipids? Emulsified, here we have fats that are lipids. If we have bile, this bile is going to separate the large fats into smaller ones. So here the enzyme lipase can come and digest easier. These are called emulsified lipids. So here are called emulsified lipids. So emulsification, it helps in the digestion process. Emulsified lipids. Okay, so these are the notes for our session.